I would like to first introduce you to Sandra Klemet-N'Guessan. I'm super sorry if I'm not able to really pronounce it correctly. Sandra did a PhD at McGill University in Montreal, Canada, where she worked on eco-evolutionary dynamics of fish movement behavior and the related conservation implications. Also of range-edge plant species in Canada, and then switched for her PhD at the Trent University in Peterborough, Canada, to animal-mediated element cycling and the role of aquatic animals in nutrient, carbon, and contaminant cycling through their excretions. And we are very, very excited to hear about what the excrements of animals tell us. So Sandra, the floor is yours.
Thank you for being here. I'm definitely looking forward to meeting all of you who I may not know yet. So I'm going to share my screen. So one little second. All right. So as Karen mentioned, the large bulk of my work during my doctoral time right now, which is coming to an end, has been on animal-mediated elemental cycling and looking at it in different contexts. So I'm going to delve in right into it by, first of all, bringing up the main topic here, which is animal elemental cycling or excretion, which often has been, or work has been focused on the nutrient part of the elements. So the reason for that is that the nutrients that are excreted by animals are bioavailable, and so they can support a large proportion of primary production. They can alleviate nutrient limitation and can also create biogeochemical hotspots. But there's been little or not as much focus on animal carbon excretion and also animal contaminant excretion. So hence come my work. And so I decided to focus on different forms of global change and how that affects animal nutrient, carbon, and contaminant excretion. The first form of global change I focused on was on seasonality and feeding ecology. And by seasonality, I mean looking at summer versus fall, and then looking at the trophic position and the littoral reliance of the animals. That is, of course, changing with climate and land use change, with seasons changing in terms of temperature intensities and, of course, precipitation patterns, but also the duration. And then the feeding ecology potentially changing because of nutrient availability in the system. The second form of global change I focused on was silver nanoparticles, and these are increasingly present in our aquatic ecosystems because they're antimicrobial agents, and so they're used in many products, including cosmetics and also in the biomedical field. And so that increased consumption by humans means that through the wastewater, they end up in our aquatic ecosystems, and organisms get to be exposed to that. The last form of global change I focused on was disorganic matter, which is also changing due to climate and land use changes. With regards to climate change, we often focus on DOM, or when we focus on DOM as units of carbon, often referred as DOC, we see that there's been an increase of DOM concentration, or DOC, especially in the northern parts of the world. When focusing on its composition, we see that in cropland-dominant areas, there's been changes in that composition. So the first part of my work was to look at the seasonality and feeding ecology effect on animal nutrient excretion in a large lake. So in this context, I use Lake Erie Western Basin as my study system. And here I'm going to briefly delve into the methods for all of my animal nutrient excretion work or animal elemental excretion work. So we would often sample different species of fish, trying to have a nice trophic position range, and also sample invertebrates. In this case here, we sampled dry cynids, but I've done also stream work where I've sampled mayflies. Then we'd be using either boat electrofishing or gill nets or insane nets, depending on the depth of the ecosystem or of the lake in this case, and then put individual fish in bags of water of a known concentration, nutrient concentration, and let them excrete, incubate them at ambient temperature for 20 to 40 minutes. And then take that water back into the lab, look at the nutrients, carbon, and potentially contaminant content of it. And we also looked at the tissue composition of, in this case, mussels and fish, looking at the stable isotopes and also just the stoichiometry of their tissue. So the questions we asked in this work are, does season affect fish and mussel nutrient excretion? Does their feeding ecology affect their nutrient excretion? And do temporal changes, meaning changes over multiple years in population biomass, affect the population nutrient excretion rates? So we expected, due to the strong effect of temperature on metabolism, that the lower temperatures in the fall would then result in lower mass normalized, meaning individual level nutrient excretion. And this is exactly what we found. The nutrient excretion rates for both nitrogen and phosphorus were much higher in the summer compared to the fall. Secondly, when looking at the feeding ecology, we expected the nutrient excretion to increase with both delta N15 and delta C13. Why? Delta N15 often represents trophic position, and we were expecting that animals that feed on prey species of or food items of higher nutrient content would then excrete more nutrients. And then in the context of Lake Erie and littoral reliance, because there is higher nutrient availability in the littoral zone of Lake Erie relative to the pelagic zone, we expected also the nutrient excretion to be higher with littoral reliance. So this was partially supported. We did find an increase in both nitrogen and phosphorus excretion rates in delta N15, but we found the opposite trend, a decrease in nitrogen and phosphorus excretion rates and delta C13 or littoral reliance. Something else that was interesting, you see here the observations are color-coded by season. And so we have the summer ones here and then the fall ones here. And so it seems like there was a seasonal effect as well with littoral reliance, with summer and individuals having a stronger reliance on the pelagic zone relative to the fall, which makes sense because in the summer, the temperatures are higher, and so a lot of fish species will go more into the pelagic zone and live there and feed there. Of course, we had more observations in the fall as well, and we see some fall observations in the pelagic zone, but this trend makes sense. And so this is now the N2P excretion ratio. So we see then the opposite trend with a decrease in the N2P excretion with trophic position, meaning that as trophic position increase, the animals excrete more nitrogen, and the opposite in relation to the littoral reliance. Now, when looking at the population nutrient excretion, we basically found that dry cynids were the highest nutrient contributors because of their incredibly high biomass. We unfortunately only had biomass data for 1 year in 2019, but when looking at then 6 years for the fish, we did see that there were changes that were basically related to the changes in biomass because we used the same individual level nutrient excretion, and that some fish species had synchronous changes in their nutrient excretion. So for example, you see gizzard shad in purple and log perch here in blue. They had a bit of a synchronous changes, likely due to the fact that they eat on different things that basically change from a year to another in opposite patterns. So in summary, we found that small-scale temporal changes, including seasonality and habitat food source, do affect individual level nutrient excretion rates. And then when looking at a larger scale and looking at the population level nutrient excretion, although we did use the same nutrient excretion rates at the individual level to scale it to the population level, it's clearly biomass does shape the relative importance of species population nutrient excretion. And so it'd be interesting to see if we were to actually measure the nutrient excretion at the individual level every year, how that would affect the overall population nutrient excretion relative to the effect of biomass. And then the last part of this work would then to be looking at how animal nutrient excretion compared to other sources of nutrients in Lake Erie. The second aspect of my work was focused on a whole lake addition experiments where silver nanoparticles were added to one lake in the experimental lakes area for 2 years. So we measured animal nutrient excretion pre and post addition, and then during the 2 years of AGNP's addition in that lake. We also used a reference lake to compare it with. And so we asked, does the exposure to AGNPs affect the nutrient excretion of the fish? And do the nutrient excretion rates, especially for the unexposed years and unexposed lakes, match model prediction based on ecological stoichiometry and metabolic theory of ecology? So these are two important theories used in animal-mediated nutrient recycling, which basically posits that certain variables affect that differently. So ecological stoichiometry posits that the body composition of the animals would have a stronger effect on their nutrient excretion rates, and metabolic theory of ecology posits that it's the body size and the temperature that will have a strong effect on the animals. And so there are models that actually combine both. And so that's what we looked at. And then the last part was to look at how the exposure to AGNPs affect the fish silver and also nutrients of silver excretion rates. So for context, here we only focused on the yellow perch as our study organism. And so we expected their excretion rates to be higher during the AGNP's exposure years because of the disruption that the exposure to AGNPs would have on their metabolism. And so that is actually not really what we found. So we didn't find any change in nitrogen excretion from pre-addition to year 2 of AGNP's exposure and post-addition. And there was no difference between the reference lake and the experimental lake. So the experimental lake is in silver and the reference lake is in black. And for phosphorus, we did find a significant increase in phosphorus excretion from year 1 to year 2 of AGNP's exposure. But again, it was not significantly different from the reference lake. And we did have a very surprising difference between the experimental and the reference lake pre-addition. So because of these unusual phosphorus excretion rates, we were curious to see if they were within a normal range or if they were really unusual even for the unexposed fish. So we used 2 models, the Shitakati et al. One, which is based on a Bayesian approach. And so you just need to plug in a lot of different variables and different values related to, again, the ecological stoichiometry and metabolic theory of ecology. And that will allow you to then simulate the nitrogen and phosphorus excretion rates and draw these confidence intervals here. So you have the 95%, 75%, and 50% confidence intervals. The Vannian-McIntyre model is more based on relationships that were drawn using a large database of nutrient excretion rates in both freshwater and marine ecosystems. And so using that, we were able to then simulate using our own values, the nitrogen and phosphorus excretion rates. So what we found is that nitrogen excretion did match for the unexposed fish model predictions for the Shitakati et al. Model, but did not match the model for the phosphorus excretion rates. So that is the case actually for both models. Phosphorus excretion rates were pretty different from what was expecting for fish of this given size that we used. But the Vannian-McIntyre model in general actually wasn't that great in predicting both our nitrogen and phosphorus excretion rates for those fish. So then when looking at the silver excretion, we expected an increase in silver excretion from year 1 to year 2 because of the longer duration of exposure to silver or nanosilver particles. So we didn't find that. There wasn't any difference in silver excretion from year 1 to year 2, but the simple fact of detecting silver excretion was already something really new and novel about our study, which hadn't been done before. So that was one thing. And then the second thing is that we did find an increase in the phosphorus to silver excretion relationship from year 1 to year 2, which is likely driven by the higher P excretion that we saw in year 2 of Agent B's exposure. So that means that fish were contributing more phosphorus relative to silver in the second year of exposure, which again could be due to some toxic effects of Agent B's exposure, but that causal effect was hard to draw. And so that's why, to summarize, we'd say that although the causal relationship is not clear, we did find that when exposed to AGNPs, fish excreted more phosphorus and did excrete silver, meaning that they would contribute to longer duration of silver resuspension in the water column after being exposed to AGNPs. So that was something really interesting. Now, the last part of my work has been focusing on looking at the relationship between ambient dissolved organic matter, both from a concentration and composition standpoints, in both streams and lakes. And in this context, I'm only going to focus on my lake work. So dissolved organic matter, or DOM, has a fundamental role in the control of an aquatic ecosystem structure, function, and services. When looking at it from a concentration perspective, or DOC, it regulates several ecosystem processes such as light penetration, thermal stratification regimes, biological productivity, and metal toxicity. When focusing on the composition aspect, it often affects the microbial activity, which will then affect how much carbon is available to other trophic levels in the food web. So in our case, again, with regards to the theory, we expected, because of the light and nutrient trade-off that is often observed in systems with higher, more or less, dissolved organic matter, we expected to then see a difference in both the biomass and the nutrient content of organisms in the system. So in low DOC environments, because of that light and nutrient trade-off, where we'd be expecting more light in low DOC environments and less nutrients, we'd be expecting a high abundance of biomass of high carbon to phosphorus content, which would then translate in a high abundance of animals of high carbon to phosphorus content. Conversely, in high DOC environments, we'd be expecting a lower biomass of primary production and lower biomass of animals, which would then and of course, lower carbon to phosphorus content, so more nutrient rich. And so then that would then affect things like the body size of the animals and also their as I just mentioned, their biomass. And since we just saw how the biomass can, of course, really shape the population level nutrient excretion rates in general, we would be expecting that the growth rates, which are tied to the body size and the biomass of the animals, would affect the nitrogen and phosphorus excretion rates. So in the context of the lake work that I did, we only focused on individual level nutrient excretion rates, but in my streams work, we actually looked at different scales, so individual, population, and community, and we're able to see how that biomass translates to changes in population nutrition excretion rates, and how that also may change or may not change with DOC and DOM. So this work was done in 11 lakes in the experimental lakes area that had a nice DOC and DOM gradient, but it did turn out that the DOC and the DOM weren't really tightly correlated. So we ended up focusing only on the DOC as our response, or as on our predictor variable. So they're basically mutually exclusive here. But we asked whether there was a relationship between ambient DOC or DOM and trophic position and fish nutrient excretion. Whether the DOC excretion rates change with ambient DOC. So in this work, we not only focus on the nutrients, but also on the DOC and DOM excretion rates. And whether fish, the fish DOM excretion signature changed with ambient DOC. So the premise of this work is, because of the light and nutrient trade-off, is really based on a unimodal relationship that's expected across trophic levels and actually functional traits. So at the basal level, we'd expect primate production to show this unimodal relationship with DOC and DOM, with an increase up to a threshold because of a high abundance of light and not as much nutrients, and then past that threshold, a decrease because of a lower abundance of light, more nutrients, but overall it wouldn't. It would end up in lower biomass, as we've seen in the previous schematic. And so we'd expect to see similar relationship for both growth rates of individuals and the biomass of the populations. And so since the growth rates would be tied to the individual level response, we'd be expecting an inverse unimodal relationship between those growth, between mass normalized nutrient excretion rates and DOC, where the nutrient excretion rates are decreasing as the body size is increasing, and then past the threshold, it's increasing because it's relative to a unit of biomass. So when looking at the nutrient excretion rates relative to DOC, we found subtle changes with DOC for both nitrogen, phosphorus, and N2P. And they were definitely not as strong as what we've seen in our streams work. So here you see both invertivores and piscivores in red and the omnivores in green. First, because we didn't have any observation in that little DOC interval. Of course, there was high uncertainty in the relationship here, especially for the omnivores. But although the nitrogen and phosphorus excretion rates did seem slightly synchronous in the way they were changing with DOC, it seemed like it was the opposite. Well, they were synchronous, but it was the opposite pattern that we saw for phosphorus. So as phosphorus excretion increased a little bit here for invertivores and piscivores, we see a little decrease in for omnivores, and then we see the opposite then at higher DOC levels. So that nice unimodal relationship we're expecting is not really what we're seeing, and that may be due to the context as well. You know, these are boreal lakes that are lower in nutrients, especially for actually phosphorus, and so that may be explaining also why that change with DOC is not as strong, although you would expect that in lakes it would be stronger than in streams because of that strong effect it has on you know, light penetration and thermal stratification regimes especially. So then using those 11 lakes, we focused on 3 of them that each represented different DOC levels. So one representing low, one representing medium, and then one representing high DOC levels. And then we looked at the DOC excretion rates and the DOC2N and DOC2P excretion rates across these 3 lakes. And so we found that fish excreted more DOC at in high DOC environments relative to medium DOC environments, and more DOC2N in high DOC environments relative to medium DOC environments. But there was no difference in their DOC excretion between low and high DOC environments, which is really interesting. That being said, so keep in mind this difference we're seeing between medium and high DOC environments. We did see the opposite difference, let's say, or pattern when looking at a DOM composition of their excretion rates. So first of all, we found that they had a unique signature in general, and that differed between different ambient DOC levels. And so in low DOC environments, we found that the DOM tended to be more aromatic and autochthonous, and that was relative to medium and high DOC lakes. So the medium and high DOC lakes didn't really differ now in their DOM composition, although they did differ in their DOC excretion, but they differ now relative to the low DOC environments. And that's likely also affected by the fact that the 2 medium and high DOC lakes were not that different in DOM composition, in their ambient DOM composition. And so that shows how the ambient environment affects what the animals excrete, but they also themselves do modify the DOM and have this unique signature.
Sandra, may I quickly interrupt you because you're already 5 minutes over time. Can you summarize quickly and finish? So
yes, I'm at the end. Yes.
Okay.
Yes. So this is it. So essentially, we found that the nutrient contribution may not change strongly with DOC, but their DOC and DOM may be different based on the ambient DOC levels. So to summarize, again, going back to that idea that I was looking at different forms of global change, we do find that changes in seasonality and the feeding ecology will affect the nutrient excretion rate through increases, especially if we expect, for example, summers to be, have higher temperatures, or just temperatures to be increased in general. We may expect to see more higher nutrient excretion rates for animals, and depending on where they feed, if that changes their feeding ecology, we may see a change as well. We expect that exposure to ADNPs will then result in excretion of silver for the fish, and the resuspension of that contaminant in the water column, meaning stronger exposure or longer exposure for other organisms in the lake. And then we do see that DOM can have a nonlinear relationship with nutrient excretion, or sometimes end up in no change. And then fish do have a unique DOM signature that changes with ambient DOC levels when exposed to DOM. So with that, I'd like to thank everybody who's been involved in this work, and of course you for listening to me, and thanks for your patience with my little longer presentation.
Thanks a lot, Sandra. Great presentation. So I guess we are quite over time, but maybe we can allow 1 or 2 questions. Is there anyone who wants to go for that? Yes, I see someone. Please, Roxanne, it's you. Go ahead, please.
Thanks, Karen. Thanks a lot, Sandra. It was really a super interesting presentation, and thank you. It was really clear. My question's actually kind of a quick one, and seems to be maybe potentially a bit unifying across your different experiments. So one thing I saw was there was definitely a higher P excretion rate, no matter what gradient you were looking at, relative to nitrogen. And where it was most apparent was in the trophic positioning of your first experiment. And there seem to be some nuances in other cases as well. And, I've thought about this, and I don't know if we know this, so I guess it's, a challenge to you to maybe disentangle, but I think it might actually be a pervasive way in which organisms in freshwaters respond, because phosphorus tends to limit primary production. So the top is helping the bottom. So I'm not sure if that's really true. But what do you think? I mean, this seems to be something that's kind of emerging. You see it like it's belling when you have more DOC, but then something else is blocking primary production then.
That's a really, really good comment. I hadn't thought about it, especially across the different studies. Certainly, yeah, in the silver nanoparticles study, that the phosphorus was really high, especially in a low P environment. That was really surprising. And yeah, I think your answer is a good element. I wonder if there's also in the case of maybe DOM and Lake Erie, if there could be an effect of just more phosphorus in the environment in general that may be also driving this.
Yeah, anyway, it's just an interesting thing to think about because the ratios are definitely changing and something's driving more P recycling, which is seems to be pervasive, which is really cool. Thanks a lot.
Thank you for your comment.
That calls for a proper and well-developed postdoc project. Awesome. Okay, given the limited time we have now, I thank you very much, Sandra. Great talk. And I would come over to our next presenter, which is Nikita Rajkumari. Nikita studied zoology at the University of Delhi in India, followed by a master graduate study on biotechnology at Punjab University in India, and now is a researcher a PhD researcher at the Indian Institute of Science and Education and Research in Kolkata, also in India. Her research interests encompass eDNA-based investigation of ecological health and state of fisheries resources with a special focus on small indigenous species of fish. And I think we will hear more about that in her talk, which we have pre-recorded, but Nikita is also here. Welcome, and you will be available for questions after the presentation, right? Okay, let's go. Thank you for that presentation.
Research, Kolkata in India. Hello everyone, my name is Nikita. I am a PhD researcher at the Indian Institute of Science, Education and Research, Kolkata in India. And I'm working with the team ITMERG. So today I'll be talking about my work, which is elucidation of biological community structure in a RAMSAR wetland, Deepor Beel, using eDNA approach, which is the only RAMSAR wetland of the state, which is located in the northeast of India. First of all, as we all know, wetlands are a very important kind of ecosystem in which, because they are being recognized globally as an important aquatic ecosystem for their role in moderating global warming and most importantly as rich repositories of biodiversity. Wetlands, they form an important fishery resource of India. So mainly a majority percentage of these area lies in the northeast of India. So these important fishery resources that I'm talking about provide livelihood and nutritional security to a huge number of fisher folks. In addition, it performs in providing many ecological functions also. So in this slide, you can see a map of Deepor Beel. As I said, it is the only RAMSAR wetland which is located in the state of Assam, a northeast state of India. So Deepor Beel, it is a perennial freshwater wetland, which is a repository of rich aquatic biodiversity. It was listed as a Ramsar site in the year of 2002, thereby recognizing it as a wetland of international importance requiring conservation. Now, this wetland is considered as one which is of international significance because of the kind of flora and fauna resources that it has. As you can see in this chart, the kind of number of species that this wetland supports, and which is also globally significant species, is quite huge. And the kind of species that I'm interested, and the study is interested in the fish species which it supports. And also, most importantly, because of its unique physiography and geomorphology, and most importantly to be highlighted, its vulnerability to anthropogenic influences makes this wetland of international significance. In this slide, I have tried to depict the kind of multitude of ecosystem services that Deepor Beel provides to the local stakeholders of this wetland. It directly sustains the livelihood of more than 800 households through fisheries, which is quite a huge number, and a total estimated fishing value which is $1,398,183 per annum has been estimated. And in this tree picture, you can see that the fisher folks are fishing for their livelihood. So the first picture depicts a fisher folk trying to check on the fish fishing net that he has kept in the last day, and trying to really check on the how much he has caught on. And the next picture shows an older lady selling those fish that has been caught from the wetland. So the fish that that is shown here, which is selling, is very interesting one actually. So I'll be talking about those type of fish in the next slide. So they are commonly called as SIS, which means small indigenous species of fish. So these are one of the ways through which Deepor Beel is sustaining the nutritional security, providing nutritional security to the people. So these are freshwater fish, and they grow to a length of around 25 to 30 centimeter maximum when they are adult. So what is the status of this SIS is that in the recent past, SIS represented an important component of the capture fisheries landings, okay, and hence it was a readily available, accessible source of micronutrient-rich food and income due to its availability. But then right now, currently, due to multiple anthropogenic threats, now it has been the abundance of SIS as compared to before has been negatively affected, and hence the price rise. It is quite expensive now, very rare to get, and hence people are not like the poor people, they are not accessible to these as their as part of their diet like before. And what is the specialty of these fish species is that they have very high profile of micronutrient content, like calcium, phosphorus, zinc, selenium, vitamin A, vitamin B12. It is very high in them, which is why it is very important to conserve them and realize the kind of nutritional security that they can provide. So in this slide, I have tried to I will be telling about how environmental DNA can be used to assess the health of an ecosystem. So environmental DNA is a short DNA fragments that the organism have left behind, maybe in non-living elements such as soil or air or water in the environment. The eDNA can be accumulated in the environment and comes from the skin, blood, saliva, mucus, eggs in the ecosystem. Like after molecular identification, it has been reported, and it appears to be more precise than visual detection. And most importantly, this method is non-invasive, which means that when samples are being collected for environmental DNA analysis, there is no harm being caused to the specimen, and which is very important. And obviously, it is a powerful tool when we use it by in the correct manner, understanding it. And also it provides a more holistic view of the biological community structure, as reported by many literature, and which is why it is a great potential for widespread implementation in the surveillance of aquatic ecosystems. And the kind of solution that this kind of technology, this kind of tool can provide is numerous, like ecological questions concerning biodiversity, diet, invasive species, etc. For example, in Deepor Beel, there has been a study which there has been a report which says that exotic species of fish which were apparently brought into the wetland from aquaculture ponds along with floodwaters are posing an additional threat to the endemic fishes of the wetland, like for example by ways of competition for food and space. And so because of all of these advantages, it makes them an excellent choice for efficient and precise biomonitoring. The objective of this study was to generate baseline information of environmental DNA dataset from the biological community of these wetlands, most importantly to aid in long-term monitoring and restoration of the wetland. So which is the first study, the first comprehensive assessment done in the whole northeast of India, in all of the RAMSAR wetlands which are located in the northeast of India. This is the first study. So this is my study site, which is Deepor Beel. I have tried to show the location of Deepor Beel and mark the sampling stations. So as you can see in the above picture, there are many boats and boatmen with them. So we actually take one of those boats and really go to each and every station that I've pointed here to collect the environmental samples and measure the in situ parameters at the site. Here is the limnochemistry of Deepor Beel. So the waters of the wetland are characterized by moderate temperature in the range of 23 degrees Celsius in winter, and neutral to alkaline pH in the range of 7.5 to 8.8, DO in the range of 5.6 to 7.8 mg per liter, and dissolved nitrate concentration in the range of 50 to 83 micromolar, which is quite high, and reactive silicate in the range of 83 to 205 micromolar, and last but not the least, total alkalinity in the range of 80 to 130 mg per liter. This is the bacterioplankton community structure. So this information is from high throughput sequencing using nanopore sequencing chemistry in a MinION platform, which was carried out to elucidate the biological community structure. So approximately 237 MB of data was generated from the environmental DNA of both the stations, and dominant bacterioplankton families were being identified. And the difference in the relative abundance of these families seen in the two studied stations are being shown in this are being represented here. And it was found that Moraxylase is the family which was abundant, was most abundant, and they belong to the class Gram-Proteobacteria. So and also here in this slide, this is the eukaryotic community structure. The abundance of Cyprinidae, which is the family of freshwater fish, were encountered in the studied wetland. So which reflects that the biodiversity of fish populations are there. And the thing to be noted is that the small indigenous species of fish that I was talking about, the SIS, they belong to this family. And many eukaryotic affiliations were also seen in the study that they said. So this is a cladogram showing the taxonomic affiliations of the eukaryotic communities found in the two studied stations, in which Eumetazoa was the most abundant. Findings so far is that high throughput sequencing results indicated the overwhelming abundance of Moraxylase, which is a prokaryote, and Eumetazoa eukaryotes. The abundance of Cyprinidae were also encountered, which reflects the biodiversity of fish populations. And according to available information on limnochemistry, waters of the wetland remained within the favorable range of water quality for fish production. However, there is a need for continuous monitoring of water quality because of continuous influx of city sewage as well as runoff from agricultural and forest lands over the years. With this, I would like to conclude my presentation, and I would like to acknowledge all the members of Integrative Taxonomy and Microbial Ecology Research Group, ITMERG, from ISL Kolkata, World Fish, and ASLO for giving me the opportunity for presenting my my work today. Thank you, everyone.
Thank you, Nikita. Thank you very much. Are you there? So we can see you live also. Hello, there you are. Good to see you. Thanks for that presentation. Do we have questions? Is there anyone who wants to know more about this? There's Roxanne. Please go ahead. Are you still muted, Roxanne?
Yeah, I know, I saw that. So I was just looking for my button. Okay, thank you very much, Nikita. I have a question in terms of the dominance of species that you saw, both in terms of the prokaryotes and eukaryotes. And, is there anything specific about those organisms in terms of their functional roles that you can describe to us, and which would explain why they help maintain the ecological integrity?
Yeah, so in terms for the eukaryotes, as I said, the Cyprinidae were abundant, and we really wanted to check that because we are saying that the SIS, they are available in these wetlands. So as predicted, it should be more, that there should be abundant, and we could see that that they were abundant. So, and also these kind of study, what is the like significance is that if there are some kind of exotic species which is not a native of the wetland, so if we are able to identify them, so then we can, in that case, which we might not be able to see by catching or something like that, but if there is some molecular signature that we get, then we can really take up some action, or we can report to the policymakers to really look into
your
their
eDNA sampling didn't see any invasives then?
No, the graphical representation that I have just showed, they are just up until the taxonomic level. So if I go into depth, maybe I can like the functional characterization. If I do, then maybe more information I can get. But as of now, it is not like it is fine at this state.
Great, thank you.
Okay. I have a question concerning the food webs. Do you know something about the food web structure, how this relates the different organisms to each other? It's a bit in the direction of Roxanne's question, but I would expect that you see changes in how the organisms are linked to each other. Do you intend to do some network analysis or something like this?
Actually, I haven't thought about network analysis, but I do look into the gut content, which is in the preliminary stage. I have not included the result, but I do look at the gut content of these SIS, the selective ones, and I do check if there are any kind of like contaminant which they are ingesting, and also to look at the diet of their like what they are eating. So I found out that they are majorly herbivorous, and also sometimes I'm able to find like traces of fish bones, smaller fish that they have ingested. And so I'm also looking into it, that if they are ingesting any anthropogenic stressed material like that.
Mm-hmm. But you don't use any stable isotopes to trace the sources?
No, I have not used any of that as of now.
Okay, thanks a lot.
There's a question in the chat. Nikita, please, can you explain
yes.
the method of sample collection?
Yeah, so the sample collection in terms of the environmental DNA, I think the question is about the environmental DNA.
I guess so.
So I collect at the sites, in the boats that I have shown in the presentation. We have marked each and every station with a GPS. We have tagged a GPS, so we follow to the exact location, and then there itself I collect the sample. For the surface water is being collected, and at the site only I fix it with molecular grade ethanol to really fix the biological community which is present at that state. And then so after this sample, this is the environmental DNA sample, and also I measure the in situ parameters at the point where I'm collecting all the samples. And after this, I take the sample to the lab, and I concentrate these samples by filtration in using a 0.22 micromole filter paper. And then after that, the biomass, the filter paper is used as a sample to extract the environmental DNA. So, and after that, that is how I collect my environmental DNA sample.
Okay, so did that answer the question? I guess yes. If not, we still have some minutes. And if there's other questions, please let us know. So I don't see them yet. And that's the moment where I would like to thank you both, Nikita and Sandra, for your presentations. Thanks a lot. It was very inspiring. And before we close, I would like to share my screen for some announcements about the upcoming Ocean Science meeting, where we have some events featured by the Raylan Cole Editorial Fellows. You can see them here. In this moment, this is about early career and student opportunities within ASLO. So we invite you here to join these events, demystifying the publishing process, all the things you want to know about writing and publishing your first paper, which is probably very important for most of you. And then also we have open access publication issues discussed. And then also us as the Early Career Committee, we have some events that might be interesting for you. Early career members the early career professional workshop, and then we have some non-academic career panels. And then also we're very happy to have another mental well-being this is a live event, a mental resilience when facing climate change and compassion fatigue. So yeah, so let's hope that we all see us there at the OSM meeting, and I wish you all a good rest of the day. Goodbye.
Thank you.